Hello everyone. So my name is Anudh Mishra and welcome to today's question and answer session. आज मैं आपके बायोलॉजी के डाउट्स कवर करने वाला हूँ. So I am a student at AIIMS Bhubaneswar right now, and let's start with the first question. The first question says, what are the types of cell wall in Monera? So the well, let's first understand the question. It is asking about cell wall. Okay, cell wall is a very important structure in a cell, and it is very important with the point of view of classification. Okay, so based on this cell wall, we have classified like kingdoms, phylums, and whatnot. Now, within the kingdom of Monera, some organisms have cell wall, some organisms do not have cell wall. For example, if we talk about mycoplasma, so mycoplasma are also members of kingdom Monera, but they lack a proper cell wall. Actually, they lack a cell wall at all. So how do they manage? They do not have a cell wall, no cell wall. So how do they manage? They actually have a rigid cell membrane structure, which actually functions as a cell wall itself, and their habitat does not glycan, which is made up of two words, peptido and glycan. So peptido means it's proteinaceous also. Glycan means it's carbohydrate also. Okay. Now, when something has both proteins and carbohydrates, what is the purpose? So proteins will actually give it a good stable structure. Okay. So if this is the cell wall, you need stability. You need stability. So proteins will give it a rigid structure, whereas the carbohydrate will give it a antigenicity or recognition. Okay. Plus, carbohydrates will also act as a glue within the proteinaceous structures. That is how a Moneran cell wall is designed. Right. So we move on to question number 2. Question number 1 is done. Next question asks, what do you mean by nucleosome and how many base pairs are there in a nucleosome? Okay, so nucleosome. First of all, you need to understand it is nothing related to nucleus, nucleolus, or anything. It is a special structure which is only formed during cell division, be it mitosis, be it meiosis. Doesn't matter. According to NCERT, it says it's there in both mitosis and meiosis. So nucleosome, what happens actually is you have something called a histone octamer. This is a histone protein. It is a protein. Now eight such proteins will form what? An octamer. Okay. And behind these four, there will be four other proteins. What your body does is it will wrap the DNA around this histone so that a lot of DNA can be wrapped and you can easily package it and transfer it between cells. Now, this whole structure is called a nucleosome. Now, one nucleosome, if you zoom it in, what does it contain? Definitely, it contains DNA. And if you zoom a DNA in, what does the DNA contain? Contains, zoom it in further, it contains base pairs. These are base pairs. How many such base pairs are there in a nucleosome is what the question is asking. So according to NCERT, about 200 base pairs per nucleosome. It is there in NCERT. You can go and check it out. Right. So I hope you understood what a nucleosome is. Right. A nucleosome is a structure which is formed only during cell division, and this structure serves as a packaging framework for what? For your DNA, and your DNA is wrapped around this. Actually, the whole thing is called nucleosome. DNA plus histone is called nucleosome, and DNA is wrapped around a histone octamer. That is how a nucleosome is described in texts, and it contains almost 200 base pairs in a nucleosome, and it has 2.5 tons of DNA coiling around it. So you take a nucleosome, you wrap the DNA once, twice, and half. That's how much wrapping is done. So 2.5 wraps. Okay, we move to the next question. It says, name a genetically engineered bacteria used for oil spills treatment. Okay, so we know that this is microbes in everyday life. We know there has been a lot of tries for making a bacteria or making a organism which can serve purpose for our human uh, race, what we want to achieve. We have a lot of genetically engineered organisms for them, be it bioterrorism, be it for the bad, be it for the good. One such example is a genetically engineered bacteria which is used for oil spills. So the name is Pseudomonas frutida. Now this is a bacteria which belongs to the genus Pseudomonas. Pseudomonas is, generally it is a pathogen. It causes pneumonia, that too hospital acquired pneumonia. So if it is hospital acquired, it is mostly antibiotic resistant. Generally Pseudomonas species, this is how they are found. They are found as pathogens. They cause pneumonia, hospital acquired pneumonia, and they're mostly antibiotic resistant. But this Pseudomonas putida, this is a genetically engineered Pseudomonas. Okay, what it does is, this whole organism is used for clearage of oil spills. Now, what is an oil spill? So imagine this is your ocean. Ocean is full of what? Water. Now a ship is going with oil. This is a tanker. A ship with oil is called a tanker. It has what? It has inside it is filled with oil. Now it was traveling from some port to another port. Somehow something happened and this oil, it got a leak, and all the oil inside the ship, spilled in the ocean. Now this oil spill is dangerous. Why? Because oil is lighter than water. It will float on the surface of water. What is wrong in that? Yes, see, there are plants inside the water, not just green leafy plants, but algae, plankton. Plus, there are a lot of fishes, other organisms, octopuses, and all. What will happen is there will be no sunlight because everything will be reflected by the oil. There will be no oxygen exchange. So the plants and animals, the biological entities in the ocean will start dying. How do you clear this? You put this one bacteria called Pseudomonas, introduce this bacteria here. This will go and clear away your oil spill. What it will do is it will go and digest all the oil and convert it into gases so that your surface becomes clear and the fish plants are able to breathe again and able to take the sunlight again. See, it has a good use. This is a good use of genetic engineering. Question number 4. What is the root? Explain primary and secondary functions. See, root is the structure which is derived or which originates from a radical in seed. You all know plumule and radicle in a seed. Root is the structure which originates from the radicle of a seed. That is the definition. Primary and secondary functions may it has to absorb water and nutrients. Secondary may we will give all sorts of functions. It has anchorage, support. It will form plant growth regulators. It will form other hormones. It will protect the plant from infections. All these are secondary functions. Clear? What is the role of microbes in the following? And I think this is an incomplete question. What are rhizoids? So we just learned about roots, right? Let's talk about a similar structure called rhizoids. Where are rhizoids seen? If you can remember, I think you can remember. Yes, you're right. Rhizoids are seen in lower plants. Lower members of kingdom Plantae do not have roots. What do they have instead? They have structures called rhizoids. And these rhizoids are root-like structures, but they're not roots. Why? Because they do not originate from a radicle. Just a second. So, because they do not originate from a radicle, why do they not originate from a radicle? Because the plants where rhizoids are found do not have a seed. Remember Pteridophyta, Bryophyta? They do not have seeds. Gymnosperms are the first plants to have seeds, naked seeds, right? Now, let's go into a bit more detail about rhizoids. What happens is, suppose this is the plant body. And here is the rock. This is a rock. And a moss has to grow on this rock. Now, how will the moss first attach itself to the rock? That is where these fine hair-like structures arise from the body. These are rhizoids. They have few absorptive and secretory properties. Mostly it's for anchorage, right? The main function is anchorage. See how the function has been reversed. Here the primary function was absorption. Here the primary function is anchorage. Secondary function is absorption and etc. Whatever. Main function is anchorage. Got it? How roots are different from rhizoids. Anything else? No. Yeah, we can move forward. Regarding alternation of generation, like polyp alternates to medusa and then to polyp again. So is it the same for jellyfish also? Okay, it's a bit, I think you're a bit confused over here. Your question asks me if alternation of generation is true for jellyfish also. Let us first understand a few terms. What is a jellyfish? We will start with the most easiest term. Jellyfish is not a biological term. It's a layman term. It's an English word for the form medusa. Medusa is a form, and that form of your cnidarians is known as jellyfish in English. So if you see a medusa in water 2000 years ago, when the English language was forming, someone saw medusa in water, a medusa form, and called it what? Jellyfish. That's how the name jellyfish comes about. So you, as a person of science, should avoid the term jellyfish. You should use the term medusa. Right? Now let us talk about polyp and medusa as a whole. This is one organism. Polyp is the fixed sessile form. Invert the polyp. This is a rock, by the way. Invert the polyp and you get a free floating form called as medusa. So you understood the difference between polyp and medusa. Now let us try to understand what is alternation of generation. Now you have a polyp which is attached to a rock. This polyp will convert itself into a medusa. Now what will happen? This medusa will find one more medusa and they will breed sexually and form medusae. These medusae turn themselves into polyps, which can again turn into polyps asexually or turn into medusae sexually, reproduce again forming medusae. So polyps produce polyps asexually, medusae produce medusae sexually. Both of them alternate between each other, and this alternation is known as, this red arrow is known as alternation of generation. Now, I hope you understood why a jellyfish, what a jellyfish is, why it should not be used, because the jellyfish is just another term for an English word for medusa. So, yes, a medusa can alternate its generation to a polyp can alternate its generation into a medusa. Now, this is a story about alternation of generation, which is often observed in Cnidaria. Now, all of the Cnidarians do not exhibit alternation of generation. There are polyp and medusa. There are some Cnidarians which are just found in polyp form or medusa form, and they do not alternate between these two forms. Examples are Adamsia for polyp, for medusa, I think Aurelia, right? So Aurelia will always stay in a medusa or jellyfish form. Adamsia will always stay in a polyp or fixed form. Got it? Okay, let's move forward. Male and female reproductive system of a frog. Okay, this is often asked, and this is given in the NCERT also. What does a male and a female reproductive system of a frog look like? I will try to draw some basic diagrams so that you get a glimpse of what it is and how it works, right? Sorry. So let's go with the male reproductive system first. Imagine we will start with a structure called the kidneys. These are your kidneys. On top of your kidneys sit the testis. Okay, I'll draw the testis in. Let's go with a brown. So this is testis. This is kidney. And now the testis and the kidney are connected by what? The testis has some sort of tubules emerging which go into the kidneys tubules. These are called vasa efferentia. They will carry your sperms and whatnot from the testis to the kidney. The kidney has its own duct, or it's not called ureter, it's called a urogenital duct. This urogenital duct opens into a rectum. So the digestive system, the last part, or rectum, but it has a secondary pouching inside the rectum. A secondary pouching inside the rectum. This is called your urinary bladder. So see, it does not directly open into the urinary bladder, but the urinary bladder is attached to the rectum where all of these things open. So a urogenital duct will carry secretions from the kidney, from the testis into the rectum. In the rectum, the GIT will send its contents, the kidney will send its contents, the testis will send its contents. So it is a three different kinds of content, and they exit the frog through a cloaca. A cloaca is a common exit for all these three secretions. This is the male reproductive system, which is pretty simple. Moving on to the female reproductive system. Again, we are going to start from the kidneys. ऑलराइट, सो सॉरी फॉर द इंटरप्शन। मैं एक बार फिर से रिवाइज करा देता हूँ मेल रिप्रोडक्टिव सिस्टम इन फ्रॉग। सो जो मेल रिप्रोडक्टिव सिस्टम है, वी विल स्टार्ट विद द किडनीज, एंड किडनीज में अटैच होते हैं आपके टेस्टिस। टेस्टिस विल कनेक्ट टू द किडनीज वाया, और टेस्टिस के बीच में कनेक्शन आता है। नाउ किडनीज से जो डक्ट निकलेगा, इन्फेक्शंस आ रहे हैं, और वो डक्ट जाके खुलेगा आपके रेक्टम में। सिमिलरली रेक्टम से एक और स्ट्रक्चर अटैच होता है जो होता है यूरिनरी ब्लैडर। सो किडनीज विल नॉट ओपन इनटू यूरिनरी ब्लैडर, बट ओके, सो ये आपका मेल रिप्रोडक्टिव सिस्टम हो गया। नाउ फीमेल रिप्रोडक्टिव सिस्टम में एक चीज डिफरेंट रहेगी, जो कि है आपका ओविडक्ट और किडनीज के बीच में कोई फंक्शनल कनेक्शन नहीं है। ठीक है, तो आपके जो ओविडक्ट होंगे वो सेपरेटली रेक्टम में ओपन होंगे, और आपके जो किडनीज के ट्यूब्यूल्स होंगे वो सेपरेटली आपके रेक्टम में ओपन होंगे। इसलिए आपके किडनीज के ट्यूब्यूल्स को हम यूरेटर कहेंगे, और ओविडक्ट तो ओविडक्ट ही रहेगा। ठीक है, अभी रेक्टम से एक यूरिनरी ब्लैडर अटैच्ड होगा, एंड दिस ओपनिंग विल बी अगेन कॉल्ड अ क्लोएका। दैट इज हाउ योर फीमेल एंड मेल रिप्रोडक्टिव सिस्टम इज फॉर्म्ड। ठीक है, तो यू हैव टू स्टार्ट विद द किडनीज, एंड देन यू हैव टू मूव ऑन फॉरवर्ड कि आपका फीमेल रिप्रोडक्टिव सिस्टम है तो ओविडक्ट और ओवरीज रहेंगे, मेल है तो टेस्टिस रहेगा। ओके, ठीक है, लेट्स मूव टू द नेक्स्ट क्वेश्चन। The next question says, how is the study of plant anatomy useful to us? ठीक है, it is a normal question, plant anatomy useful क्यों है? तो सी प्लांट एनाटॉमी से ऐसे हम लोग बहुत स्टडी कर चुके हैं, एंड देयर इज नॉट मच लेफ्ट टू स्टडी इन दैट, बट स्टिल व्हाट इज प्लांट एनाटॉमी? फर्स्ट, प्लांट एनाटॉमी इज द स्टडी ऑफ इंटरनल स्ट्रक्चर ऑफ प्लांट। प्लांट के अंदर क्या चल रहा है, कैसा स्ट्रक्चर है, इफ वी नो दैट, then only we'll be able to figure out how to use it to our advantage. Like how does photosynthesis work? If we find a clue to that, how does the plant survive stuff? How does the plant protect itself? How does the plant transport its food? We find a lot of insight into how living systems work, how physics is coming into action in biology, how chemistry is coming into action in biology, how the structure is arranged. A lot of applications are there. Like studying plant anatomy helps us to decode how years of evolution has turned the plant into this, and how we can use it to our advantage and build structures and whatnot. Right. So yeah, that is the answer. Moving to the next question. Define Sertoli cells. Now, this is a human reproductive question. We will draw the seminiferous tubules. If this is a seminiferous tubule and I take a cut section, this is how it will look. The lumen will be lined by germ cells. Now, this is germ cells, and inside the lumen will be filled with sperms. This is a male reproductive system, right? A Sertoli cell is a cell which is between these sperms so it forms a boundary. Look at this boundary. appreciate this boundary formed by these Sertoli cells. The purple cells are, these cells are Sertoli cells. The Sertoli cells are found in male reproductive tract, and they form a boundary between the blood and the sperms. So the blood, it cannot enter and destroy the sperms because of this boundary. That is what the main function of Sertoli cell is. Other than that, a Sertoli cell has other important functions. Form blood, testis, barrier. Otherwise, what will happen? This is a sperm. A sperm is 23 chromosomes. So it is not a normal cell. If it is not a normal cell, your immune system in the blood will go and attack the sperm, which we don't want. That is why we form a blood testis barrier using the Sertoli cells. Right? The next thing, Sertoli cells secrete androgens. They help in nutrition of sperm. All these are the function of Sertoli cells. Okay. Next, we have digestive system of frog. Again, frog comes up. Very simple digestive system. You don't have to stress much. It will start with the mouth and buccal cavity. This will lead it to a pharynx, which will go into the esophagus and stomach. Now, in the stomach, unlike our stomach, the digestive system of a frog is a bit different. What happens is, there is this structure called liver and gallbladder. Okay, they kind of pour the secretion into the stomach rather than the duodenum. Liver and gallbladder will pour their secretion into the stomach. After the stomach, you will have the small intestine. No duodenum, ileum, jejunum. It is just intestine. It is just the small intestine. Okay, pancreas. Again, pancreas will pour its secretion into the stomach, which will all go into the intestine. The intestine opens up into rectum, which opens up into cloaca. That is how the whole digestive system is organized. To sum it up, it starts with your mouth. Mouth opens up into the pharynx. Buccal cavity. The buccal cavity opens up into the pharynx, the esophagus, which goes into the stomach, where digestive juices from liver, gallbladder, pancreas, and all are secreted. Further, the food moves into the small intestine, goes to the large intestine or the rectum, and opens outside via the cloaca. Understood? Question number 12. What is dorsi-ventral? So, question number 12 is dorso-ventral. Now, dorso-ventral is, see, dorsal and ventral. So, if something is dorso-ventrally flattened, dorso-ventrally thickened, which means that if this is the organism, you see the dorsal, and underneath it is the ventral side. So if I draw it like this is the dorsal side, this is the ventral side. This is the axis we are talking about, dorsiventral axis. If it is thickened in the dorsiventral axis, means it is thickened in this axis. If it is bulged in the dorsiventral axis, means it is bulging in this axis. That is what dorsiventral means. Dorsal ventral axis, the axis from dorsal side to the ventral side, is known as Dorso-ventral axis. Next question. Ammonia, urea, uric acid, which is most toxic and which is mostly soluble? Where does metabolism of ammonia take place? So among your three excretory products, this is the order of toxicity. Ammonia is highly toxic. If you store, you cannot actually store ammonia in your body. You cannot actually afford to keep ammonia inside your body. What will happen? Ammonia forms basic radicals, ammonium radicals, and it will try to, destroy all your body tissues. It will go into your brain. It will cause encephalopathy and all not whatnot. You cannot afford to keep ammonia in your body. Plus, ammonia requires a lot of water for its excretion. So in terms of solubility, ammonia is not that soluble. Why? Because it needs a lot of water for its excretion. Requirement of water. Again, this order. But if you reverse it, that is water required for excretion. Uric acid is, very, it is very less toxic. Plus, it requires the least amount of water to be excreted. So that is the order. Where does metabolism of ammonia take place? So ammonia is converted into urea in our body. If you're talking about human body, it takes place in liver. So this is the liver. Here ammonia will come and it will convert into urea by urea cycle. So urea cycle, ornithine cycle, Foris cycle, all these cycles, they will convert ammonia into a less toxic form called urea. Now this urea is going to be excreted in the kidneys. Okay, the kidneys will take up the urea, throw them out of your body. Liver will again convert ammonia into urea so that it is excreted by the kidneys. That is how all of it works. What is Archesporium and where is it present? See, Archesporium, it is all primitive reproductive structures of plant. Just like the plant, the gymnosperm angiosperms have oogonia and the pollen grains. Archesporium is very primitive. It is present in plants like Peridophyta, Bryophyta. If you're talking about Archegonium is also present in gymnosperm, apart from Peridophyta and Bryophyta. That is Archegonium, but we are talking about Archesporium. It is only present in Pterido and Bryophyta. What is the function of Archesporium? It will produce your four mother cells. Okay. And they will produce spores, microspores. Okay. Next question. What is osmolarity? So the term actually means osmolarity. Osmolarity is a concentration term. It is expressed in osmoles or milliosmoles. This is the unit. And if I take a beaker full of water and dissolve some solutes in it, comparing it to another beaker and I dissolve a lot of solids in the similar amount of water, the osmolarity here will be really greater and more than the osmolarity over the first beaker, which means osmolarity denotes the amount of solutes dissolved in a solution. That is basically what it does. It will tell you how much osmotic pressure a particular solution is going to exert over a pure water body. So what happens is when you give a a solution in which a solute is dissolved in the solvent, give it with pure solvent, this solution will exert a pressure on the pure solvent. Why? Because you are separating both of them. I'll show you it in a diagrammatic form. What happens is this is a solution and you are separating it from a pure solvent. Now this separation will try to exert a pressure over the pure solvent. Why? Because these particles are moving. These solute particles are moving here and there, and they want to enter this free zone because there is no solute over there. They want to enter in this free zone. That's why they push this membrane between them. Now, this push creates a pressure. This is known as osmotic pressure. Osmolarity can be also defined in terms of osmotic pressure. As the more osmotic pressure it is exerting, the more the osmolarity is there. But in general, you can call it it denotes the amount of solute dissolved in free solvent. Okay. What is tissue and difference between animal and plant tissue? So tissue. It's a very basic question. What a tissue is? A group of cells. A group of cells similar in origin and function are called tissue. Now, two criteria: origin and function. They should be of the same origin. They should perform a similar function. Then only the cells will form a tissue. A tissue में जो cells होते हैं, they will have similar origin. They will perform similar function. Example is your heart tissue or myocardium. Now, myocardium, all the cells are originated from mesoderm, and their function is contraction, relaxation, which achieves what? Pumping of blood. That makes them a tissue, right? Group of cells, similar in common in origin and similar in function, form a tissue. Got it, guys? Why penicillin does not work on mycoplasma? Okay. So what is penicillin? First of all, what is mycoplasma and why does penicillin not work on mycoplasma? Penicillin, as you all know, it is an antibiotic discovered in the fungi called Penicillium by Alexander Fleming. This penicillin is an antibiotic. What does this penicillin do? Actually, the penicillin stops cell wall synthesis. Okay, that is the primary goal of penicillin. It will stop the cell wall synthesis in a bacteria. What will that achieve? You stop the cell wall synthesis. The bacteria cannot get a cell wall. It cannot stop substances from going inside it. It cannot help. It cannot protect itself. It will die. Penicillin kills bacteria this way. Now, mycoplasma lacks a cell wall. Mycoplasma, there is no cell wall at all. How will penicillin go and act on it? Right, you're right. Penicillin will not act on mycoplasma. The simple answer is because mycoplasma has no cell wall and penicillin stops cell wall synthesis. These two cannot be clubbed. You cannot give penicillin to kill mycoplasma. Mycoplasma doesn't care if you're giving penicillin or not. It has no cell wall. Even if you give penicillin and try to stop cell wall synthesis, doesn't matter because it has no cell wall. It won't be forming cell wall. It won't be having all those metabolic processes required to form cell wall. So mycoplasma is totally unaffected by your penicillin. You will have to give some other antibiotic which acts on some other mechanism, another metabolic pathway of a bacteria of a Monera to stop a mycoplasma from growing. How GFR rate increases? That is the question. So, firstly, the full form of GFR is glomerular filtration rate. So, there is nothing called as GFR rate. Okay, unless and until you define the rate of a rate. GFR is glomerular filtration rate. What is it? Suppose this is the Bowman's capsule. This is PCD. This is Bowman's capsule. Here comes your afferent arteriole from the glomerulus. And here goes your afferent arteriole, taking the blood away. What happens in glomerular filtration is because the afferent arteriole diameter is larger than the afferent arteriole, you're basically pushing blood and you're treating a pressure difference. This pressure difference is forcing fluid into this Bowman's capsule, into the PCD. How can you increase this filtration rate is, yes, you guessed it right, by altering this pushing thrust. You know, I told you about the push, right? This push, which is getting generated because of difference in the radius of efferent and afferent arteriole, can cause a significant increase or decrease in GFR. How can you increase the GFR? Simply increase the push. How can you increase the push? First thing, increase the blood pressure. The blood pressure, increase the blood flow. Both of these will help more blood into getting filtrated. Third thing is either you decrease the efferent arteriole ka radius or increase the afferent arteriole ka radius. If you do both of these things in a balanced manner, like if you increase the afferent arteriole too much, then also blood will not find adequate pressure. But if you just increase it enough and decrease this one enough, there will be increase in this pushing force, right? So it has to be done in moderation. Okay. That is how you can increase the GFR. Next, 19 is what is a germ layer? Now, this is plant diversity question. When a zygote is formed, it will form tissues. These tissues will be arranged. These tissues before getting formed will be arranged in clusters of cells. This is the cluster of cells. So in the very primitive organisms, there were only two germ layers. Actually, their germ layers are layers because I will start with the gut. This is the gut. Around the gut, there will be one germ layer. Around that germ layer, there will be another germ layer. So this inner germ layer is endoderm. Outer germ layer is ectoderm. In humans and advanced animals, the gut and endoderm and between the ectoderm there is a third germ layer. This is the endoderm. This is the ectoderm. This is the third germ layer. Of course, it is called mesoderm because it is in between. This mesoderm forms structures like your muscles and other bones and all. Ectoderm and endoderm have their own structures. Mesoderm forms its own structures. Now, in pseudo coelomates, true coelomates and coelomates, what is that concept? So, coelomates, what happens is this is ectoderm, this is the gut, this is the ectoderm, this is the endoderm. Now, this mesoderm inside is arranged in pouches. Not pouches. The whole body cavity is lined by mesoderm. See, the body cavity is lined totally by mesoderm. This is your coelomates. This is called a coelom. Pseudo-coelomates, we'll first describe acoelomates. Acoelomates, there is just two layers. There is no body cavity. Endoderm, ectoderm. Now, pseudo-coelomates में क्या होता है? Even in coelomates, the mesoderm, if it's there, there is no body cavity. Pseudo-coelomates में there is an ectoderm, there is an endoderm. The mesoderm is present in pouches. This is mesoderm, and there are small cavities lined by mesoderm, and they are present in pouches. That is pseudocoelomate. So these small cavities are called pseudocoelomes. Okay. Let's go to the next question. Difference between red algae and brown algae. Firstly, red algae. Okay, brown algae, red algae. Brown algae, fucoxanthin is the main pigment. Red algae mein you have phycoerythrin. Brown algae can be huge, like kelps, huge bodies. Red algae are not, they do not form huge bodies. Brown algae mein your cell wall is made up of cellulose, hemicellulose, tin, right? Red algae may be cellulose, hemicellulose, pectin, sometimes calcium carbonate. Okay. Chlorophyll A, chlorophyll C. But brown and red algae में chlorophyll A, chlorophyll D. Okay. Brown algae में you have flagella. Red algae no flagella. Okay. Brown algae live in moderate depth. Red algae live in great depths. And there is a table in NCERT which you have to learn. Go and learn that table. Okay. There is a table which you know, I'm talking about this table at the towards the end of your algae chapter topic. इसमें red, green और brown algae का difference दिया हुआ. So you should go and read that table which is given in NCERT. Okay. Cellulose. So this is a point which I missed. So brown algae mein, which is made up of cellulose and algin. Red algae mein cellulose, pectin, and polysulfate esters. Okay. One more point I missed is stored food. Stored food in brown algae is mannitol. laminare. And red algae, it is Floridian starch. Okay. Rest all flagella points and all that I have covered. That is question number 20. Let's move to question number 21. Again, how is the study of plant anatomy useful for us? I'll skip this question. 22 says difference between mitosis and meiosis. Mitosis में daughter cell is equal to parent cell. Exact copy. Meiosis में daughter cell is not equal to parent cell. Mitosis में chromosome number equal or preserved. Meiosis में chromosome number half. Okay. Mitosis में no change in genes. Meiosis में crossing over, recombination. So the cells are different. Even daughter cell is equal to daughter cell in mitosis, but daughter cell is not equal to daughter cell in meiosis because of genetic information. Mitosis is seen in commonly in epithelial cells in our body, and meiosis is seen during reproduction, so reproductive cells. Content of chromosome are same, but different amount of Q and A are double. Okay. Amount of DNA. Okay. Content of chromosome are same, but amount of DNA are double. There is a question, it's an incompletely formed question, saying that chromosome content is same, but DNA content is double. How is it possible? Maybe that is how. That is what it is asking. So let us take a cell with 2 chromosomes. This is a cell with 2 chromosomes. Interphase में क्या होगा? Interphase में this cell will be all the chromosomes. Okay, I'll draw the 2 chromosomes in different colors, blue and black. So interphase में the DNA will be decondensed. As soon as you reach your mitosis, before mitosis also, what you will do is you will replicate the DNA because you have to distribute the DNA into 2 cells. Now what will happen? Double the amount of DNA. See, this is more dense. Has double the amount of DNA. So if DNA का content इधर पे Z था, इधर पे 2Z हो गया है. But what happens is, if you look closely, I'm drawing it here. If you look closely, this is DNA 1. This is DNA 2. These are same DNA because this has been replicated from each other. But when you turn it into a chromosome, this is chromatid 1. This is chromatid 2. They are attached at a centromere. So technically, this is only one body. Okay, this is one body, and 2 such bodies are present, which means at the centromere it is attached, and 2 such bodies are present. This is chromosome 1, this is chromosome 2, just exactly as it was in the parent cell. There is no difference between these. That is how you say, although the DNA number has doubled, but the chromosome number is still the same. All right. So that was all the questions for this session. I'll see you in the next session. Till then, take care. Thank you and bye.